This is part 5 of our Chapter 9 lecture. So finishing up our citric acid cycle and oxidative phosphorylation, we have ATP synthase forming our worker protein to make ATP through chemiosmosis. So oxidative phosphorylation sets up a proton gradient in the intermembrane space of the mitochondrion. And as that gradient is created, it allows or actually encourages the flow of hydrogen protons through the ATP synthase, which then that energy is used to power the bonding of inorganic phosphate to an ADP molecule. The hydrogen proton gradient is referred to as a proton motive force. So if you see that word, you'll know that simply means the gradient that is being set up with the buildup of a high concentration of hydrogen protons in the intermembrane space, which is setting up this potential energy, this capacity to do some work, which will be eventually the synthesis of ADP and inorganic phosphate into ATP, or actually the phosphorylation of ADP. So, counting up our ATP produced by cellular respiration, we need to think again about the whole process. So we have in cellular respiration the breakdown of glucose, which step by step is allowing the reduction of NAD to NADH, so that now NADH is an electron carrier which will take the electrons to the electron transport chain, which then, as the electrons pass through that chain, we have a proton motive force being established, which then is going to produce our ATP. About 34% of the energy in a glucose molecule ends up being transferred to ATP during cellular respiration. So we get about a third of the energy that is potentially in the glucose molecule being transferred, and we end up with about 32 ATP. The rest of that energy is going to be mostly lost as heat. This is a diagram that shows that where we get our different electron carriers and where we're also getting our ATPs out. So looking at the electron carriers from glycolysis, we start with our two NADHs, which are going to cross the membranes of the mitochondria and enter into the matrix here. And we'll talk about this in a second. We also have 2 coming out of the oxidation of pyruvate, and we have 6 coming out of the citric acid cycle, and we have 2 FADH2s also coming out of the citric acid cycle. So for each glucose molecule, we're going to produce 10 NADHs and 2 FADH2s. And those are all going to be available to help oxidative phosphorylation occur and the chemiosmosis of ATPs. So we end up with about 26 to 28 ATPs. They have a range because they actually don't know specifically what causes there to be a difference, but there is a difference, and it varies from cell to cell. But we have this as our general figure, 26 to 28 ATP being generated by oxidative phosphorylation and chemiosmosis here. Through substrate level phosphorylation, we have 2 ATPs being made by glycolysis and then 2 coming out of the citric acid cycle. So these 4 and these 26 to 28 are how we end up with a total of 30 to 32 ATPs per glucose molecule. This is showing how these 2 NADHs coming from glycolysis break down to pyruvate. When we have these electrons, shuttles spanning the membrane, the NADHs can actually take one of two shuttles, and one's going to end up carrying the two NADHs into the mitochondria, and the other is going to take the electrons and attach them to an FADH2. So we actually could end up with either of these as a result of glycolysis. But to make things simple, just think of glycolysis as producing two NADHs. So this picture also shows our overall process of the oxidative phosphorylation. We have glycolysis out here making our pyruvate, but then we have our Krebs cycle, which is together, these are harvesting electrons, which are then going to be carried by our electron transport molecules to our electron transport chain. The electron transport chain is going to provide the energy, or it's actually going to cause the flow and accumulation of hydrogen protons in the intermembrane space. Then we have oxygen as our final acceptor down here, waiting for these electrons. And so as the electrons are drawn down the electron transport chain, they come and join the oxygen atoms here, which immediately join with hydrogen protons to make water. Then we have this proton motive force, this buildup of hydrogens, and they are then going to flow through our ATP synthase, so we get our production of ATP. So quickly, I want you to try and answer this question. Which of the following statements comparing substrate level phosphorylation and phosphorylation by chemiosmosis is incorrect? Stop the video and answer the question. And I see we're missing part A. But the correct answer is actually A. So it should not have been absent. So you didn't even have the right answer to choose from. But the answer is that both of these, the one that's not correct about these, is that both do not involve accessing energy in the form of an electrochemical gradient across a membrane. Only chemiosmosis uses that electrochemical gradient. Substrate level phosphorylation, which is how we get ATP from glycolysis and the Krebs cycle, relies on a breakdown of a molecule and getting the phosphate from a organic molecule to actually add to an ADP molecule. So it's an actual chemical reaction as opposed to a proton gradient. So our final section, 9.5 in the textbook, is on fermentation and anaerobic respiration. And these are important to know because we don't always have oxygen present, so cells need another way to get some ATP and to get energy to do their functioning. So most cellular respiration is going to require oxygen to produce ATP, and without oxygen, the electron transport chain does not work at all. So in those cases, glycolysis can still occur, but there has to be another process, and those are the anaerobic respiration or fermentation to produce ATP. So we have glycolysis occurring and producing pyruvate, whether there's oxygen present or not. And what happens to pyruvate is then going to be determined by whether oxygen is there. So if there's no oxygen, the cell is going to start the process of fermentation or anaerobic respiration. And if there is oxygen, then the pyruvate will enter the mitochondrion and the Krebs cycle will perform aerobic respiration. So without oxygen, we have anaerobic respiration, which will use an electron transport chain, But in that case, we don't have oxygen, so the final electron acceptor is going to be some other molecule like sulfate. And in fermentation, we use substrate level phosphorylation instead of an electron transport chain to generate ATP. And that substrate level phosphorylation is specifically glycolysis. So fermentation has glycolysis plus reactions to regenerate the NAD plus molecule to be reused. And we have two types of fermentation. We have alcohol and lactic acid fermentation. So these are our two types. You can see here we have glycolysis producing our pyruvate. And then instead of going into the Krebs cycle, the pyruvate goes into two more steps. It goes through two steps with an end product, either ethanol, a form of alcohol, or lactate in lactic acid fermentation. So these are our two different types here. In alcohol fermentation, the pyruvate's converted to ethanol in these two steps. So in this case, with alcohol fermentation, we do get carbon dioxide as pyruvate is broken into acetaldehyde. And in that process, we have NADH being formed. But in glycolysis, we do have NADH being formed. But if that continues without the recycling of NAD+, we will no longer have something for these electrons to be added to in order to form the NADH. And then we will no longer have glycolysis continue because this will just stop without this molecule present. So there has to be a recycling that occurs. So what we need to have occur is the oxidation of NADH back to NAD+. And this occurs in our second step of fermentation and the production of ethanol. In lactic acid, we have the same problem. We need to get the regeneration of NAD in order for glycolysis to continue. And so in this case, we have pyruvate going directly into lactate. In that process, oxidizes our NADH back to NAD+, so that, again, we have the basic molecules needed for glycolysis to occur in the breakdown of glucose. And all of this is needed because we are trying to get some ATP out for the cell's use. This lactic acid fermentation is used by some fungi and bacteria. It's used to make cheese and yogurt. But we also are probably even more familiar with it when we don't have oxygen present or when the oxygen in our cells has been used up, particularly in our muscle cells. So in some exercise, if you've done something that uses up your oxygen really quickly, and you continue to have a demand on those muscles, you'll start to feel some pain. And that pain is actually the buildup of that lactate in those muscles because the cell's still trying to produce ATP, but it doesn't have oxygen for the pyruvate to be drawn into the Krebs cycle. And so instead, it's quickly trying to get some more ATP by shunting the pyruvate into this lactic acid fermentation. This is a quick overview, but you can see this on the Pearson website because I want to end with a comparison of fermentation with anaerobic and aerobic respiration. So all glycolysis uses, all these processes use glycolysis to oxidize glucose, and all three, NAD+ is the oxidizing agent. And the processes do have different mechanisms for oxidizing NADH. And you can read through this quicker than I can say it. So pause this and read through it, and then I'm going to show you really quickly the last slide so you can pause and take a look at this one as well. And this is the final information for you to be familiar with for this chapter.